Today we're incredibly fortunate to have Adit Radha here. He's an assistant professor at MIT in the Applied Math Department, and he's also an associate member of the Broad Institute at MIT and Harvard. He was a postdoc fellow previously at the Broad Institute at the Eric and Wendy Schmidt Center. That's actually where we met. We were both postdocs there. And before that, he was the George F. Carrier postdoc fellow at Harvard. He got his PhD in electrical engineering and computer science at MIT, and he's really done some super interesting work on trying to understand what neural networks are doing, how they're doing what they're doing. What I really appreciate about Adit is the way he explains very, very difficult concepts in a really clear, nice, easy to understand way. It's really helped me actually understand what neural networks are doing too. So I'm really excited to hear his talk. Hope you guys are too, and we'll let him take it up.
Thank you, Bijan, for the. Thank you, everyone, for coming, and thanks for the very kind introduction. So yes, I will be talking about how we've been approaching this problem of understanding how neural networks learn features or patterns from data. And just to begin, I actually wanted to talk about the prevalence of sort of high-dimensional data today. And the two applications and common threads I have running through this talk is comparing large-scale text databases and sort of the explosion in text data that's available. So today you can just go and download data sets of millions and billions of documents. And why is this data high-dimensional? The idea is that any given passage of text I can typically encode into the form of a matrix that represents what tokens are occurring in which position and so on. And typically nowadays, at least as of a few years ago, language models were using, effectively 100,000 dimensional context lengths and vocab sizes of around 100,000. So here's some examples. We'll actually return to open web text where you have 8 million documents. This is just all readily available for download for people to just work off of. On the other hand, while this has been going on and these collections have been going on very recently, we have a sort of explosion in the biomedical world, and especially the area I work in is related to genomics, where we have this explosion in single-cell RNA sequencing data, where we now have orders of millions of sequence cells. We'll see an atlas where we have 30 million sequence cells come up later. And the idea here is that a cell is represented as a high-dimensional vector, so 20,000 dimensional vector, where each entry is the counts of a gene. And so now we have on the order of over 100 million cells. There's initiatives to actually get 500 million cells and a billion cells and so on. And so what are we doing with all of this data? There's really two big opportunities here. One is on the predictive modeling side. So for example, language modeling, you can do training objectives, where here you're just predicting, let's say, the next token given a context of previous tokens. But you also have the opportunity beyond necessarily predictive modeling to actually discover underlying structure and patterns in data. And why does this really matter? At least in the biomedical side, if I look at single-cell RNA sequencing data, you can start to identify patterns in gene expression that lead to new biological discoveries. And in fact, this is ultimately what leads to targeted therapeutics for diseases. An example would be, gene therapy for sickle cell very recently. So what do we want out of these opportunities? We really want fast and effective algorithms for solving these problems. And what do we have today? Well, on language, I hope it's very clear to many of you, we have large language models which are incredibly effective. And really the pre-training procedure for language models is this idea of next token completion. Yet you can actually see that the inputs to these systems are incredibly high dimensional. So if I was to look at the space of all possible tokens and vocab that are going into this thing, I have 100K by 100K matrices that I could populate. This is just an insane amount of data that could exist. Yet what we see is that despite the high dimensionality, these models are performing exceptionally well. And they've kind of revolutionized a lot of technology today. So the key question has been, what makes neural networks so effective? And there's a hypothesis in the field. The hypothesis for many, many years, even going back to the early papers by Hinton and so on, you'll see that they claim neural networks are learning low dimensional representations or features of data. And This has now become an extensive area of study in the theoretical side, but there's really two sorts of fundamental questions here. The first one is, how does feature learning actually happen in neural networks? What does it mean for feature learning to happen? The second one is, how does feature learning identify low rank or low dimensional structure in data? And the two applications I'm going to talk about, we're going to talk about, how we began to answer these questions, and then talk about two major applications of answering these questions. The first one is going to be on steering large language models, and the other one is going to be on the biomedical side, discovering cellular programs through feature learning. And this is effectively the roadmap to the talk, so I'm going to talk about our work first on how does feature learning occur in neural networks and what our hypothesis is around this. So I want to start with a very simple example, but one that really clearly illustrates what feature learning is and why it's interesting. So let's start with a very simple case of a supervised learning problem, where I just train a neural network to classify whether someone is wearing glasses in image data or not. And so what data set am I going to use? I'm going to use the Celeb A data set. This is supposedly a data set of celebrities, although I think out of all the times I've given this talk, one person has maybe identified a celebrity among these images. But the idea is that these images are centered, so the eyes are always in the center of the images. What I'm going to do is there's 27,000 pixels roughly in these images. I'm just going to flatten them into vectors and do a really simple model, which is just a two-layer feed-forward nonlinear network. And the exact architecture of the network is here. I have a vector going in. I have matrices interspersed with ReLU activations. And I just at the end make a prediction whether someone's wearing glasses or not. This is really simple to do. There are standard libraries to help you do this now. You're going to see that the neural network gets very high accuracy on test data. So above 90%. But the real question is, what are the features that are learned? And there's a very simple visualization to start to look at what features are learned. What you can do is you can effectively look at the covariance matrix, the uncentered covariance matrix, which we call the neural feature matrix of the first layer weights. So this is just W1 transpose itself. And the idea here is that the eigenvectors and the eigenvalues are capturing the singular vectors and values of this matrix W. So what I can do is I have a 27,000 by 27,000 matrix. I have one value for every diagonal entry here. So I have one value per pixel in my image. So what I can do is reshape this diagonal into an image and visualize it through training. And if I do that, I start with a randomly initialized network. This picture sort of looks like random noise. But as I go through training, what you'll see is that the model actually just learns to predict a filter on the diagonal looking for glasses. And why is this surprising? The reason it's surprising is when you train this model, you just train it with gradient descent. You don't impose any sort of regularization. You don't impose any sort of other objective to force sparsity on the first layer weights or so on. Nevertheless, you're getting it. So I don't know of any other model at the moment that actually has this sort of property built in without an explicit regularizer. And the key here is that neural networks are really learning to throw away all the pixels that they deem not relevant for this predictive task and effectively restricting to 1% or fewer of the pixels that matter. So the question is, how are such features actually emerging? Well, if we actually take a step back and ask a broader question, given any predictor f, how would you identify the important features? Well, if I only have access to the input and output, what I could do is something, again, very simple. I could perturb each of the features a little bit and see how the output changes. And the idea, the key idea here would be that important features are those that most change the predictions when I change them a little bit on the input. And if I'm looking at change in output by change in input, this is a gradient, this is a derivative. And in fact, instead of looking at just coordinates individually, it turns out there's a statistical operator that you can look at for measuring feature importance, which is called the average gradient outer product, or AGOP. And in fact, this goes back quite early in statistical literature. But the idea of the AGOP is that given any predictor f and data points x1 to xn, you take the gradient of f with respect to the input data. This is a vector. And I take that vector and its transpose, and I average over all my data points. Now, this is a nice, simple method for identifying important features. But the real claim that we have is that neural networks are implicitly implementing this procedure through training. And what I've stated here is what we've posed as an ansatz, which is that the covariance stated only for the first layer here. The covariance of the weights at the first layer are element-wise proportional. This is a matrix, so it's element-wise proportional to the AGOP of my trained neural network raised to some power. And I've written it here for the first layer, but in fact, this extends to deeper layers. But now instead of computing the gradients with respect to the input data, you would compute it with respect to the input of any given layer. What evidence do we have for this ansatz? It turns out that it's exactly true in deep linear networks. So when you don't have any nonlinearity, this uses a theorem. Interestingly, that has some implications for matrix factorization and low-rank matrix completion that I won't go into today. But what I will go into today is the empirical evidence. And in our paper, we show a bunch of empirical evidence on fully connected networks, convolutional networks, and transformers. And so that's what I'll show. So just for some empirical evidence in fully connected networks or multi-layer perceptrons, what we did here is actually just train 5 hidden layer, with 1024 MLPs. We trained them across 121 different data sets. And for each of these 121 different MLPs, at every layer, we look at the correlation between the AGOP and these neural feature matrices, these covariance matrices at any given layer. And what you'll see is that the correlation between them is exceptionally high. It's always on the median above 0.95 or so. So these two things are almost actually proportional, and in many cases, you'll see that they're actually just numerically 1. What I'll talk about quickly here is how does this ansatz work for convolutional networks? And what is a convolutional neural network? Well, you'll typically see some kind of diagram that looks like this, which is not very helpful. But the idea is that you have an image going in, you have a label coming out. But really, the proper way to view it is that convolutional networks are actually functions on patches. And so what I can do is I can actually write convolutional nets as a function of the form f on m patches from an image, and I'm really multiplying each patch by a weight matrix. And so the ansatz, what it says here, is that this shared weight matrix across patches is proportional to the gradient outer product of f with respect to the patch level information. So the gradients instead of inputs are with respect to patches. And so when you have an ansatz like this, you can start to ask, what are the features that are learned on patches, and does this ansatz hold in realistic convolutional neural nets, things that I didn't train? So in fact, this is what we wanted to analyze. So what we did is we took state-of-the-art pre-trained convolutional nets as of 2016-17. These are all trained by industries, so things like ResNet is going to be trained by Microsoft. VGGs were a very popular model, and we have AlexNet as well. And what we did is these are all trained on the dataset ImageNet, which is about 1 million different images where you classify among 1,000 objects. And you can look at every single layer. So here, each column here is a model. And the y-axis here is just the Pearson correlation between my covariance of this weight matrix at the patch level versus my AGOP. And you can see here that the correlation is actually...
No.
Oh.
So... Okay, so whatever you can go. What I was thinking, I looked at maybe week October.
Can someone,
maybe.
No, I think it's,
Friday,
this needs
all day Thursday to do it. October 6th. My hair is October 4th. 3rd. October 3rd. 3rd, okay, good. Yeah, this week is wide open. The week after we have talk. Okay, yeah, anytime. Yeah, let me see what I.
Is there any way to mute that? Yeah.
Okay. I get the cheapest price. Yeah.
Okay, cool. Thanks. Okay. All right. Okay, anyway, so going back to this, the idea here is that between the AGOP and NFM of any given weight matrix, you'll see that there's incredibly high correlation once again. And in fact, you'll see that the variance of these correlations starts to grow higher for poorly performing models. So the models on the left perform better, models on the right had worse performance. But now you can start to ask, what are the features that are actually learned by these models? Well, what you can do, and I'm going to show an example here for VGG19, where you can compute this AGOP. The AGOP is a patch level. You can think about it as a filter on patches or an operator on patches. So I can take any image, I can take a patch of that image, multiply it by the AGOP, and then take its norm. This is a vector. I take its norm and visualize that across all patches. So I took VGG19, I computed this AGOP, and what you'll see in the first layer is that the AGOP actually just does edge detection. So this is something, and if you look at the eigenvectors, it's a horizontal and vertical edge detector. So what you'll see is that the AGOP of this trained model is doing edge detection. As I go deeper and deeper, you'll see that it starts to do a progressive sort of edge detection and ultimately ends up highlighting regions of the object that are important for classification. So for example, for dogs, it's the eyes of dogs that end up becoming the key feature, key spatial feature that's important. For the ladybug, it's this region of the ladybug. And for, strawberries, it's that region of the strawberry. And the reason things are getting blurrier and blurrier is because the model is downsampling the image through the layers. Okay. So now that I've sort of stated what this ansatz is and my claim about feature learning in neural nets, what is the purpose of this? If you already have a trained model, there's no reason to compute AGOPs now and sort of use that to just, verify the features. The real power of the AGOP is that it can be computed for any function, not just neural networks. It's just a general operator that takes in a function and data points and gives you a matrix. And so what you can do with it is you can construct an algorithm for feature learning with any predictive model, not just neural networks. And what you can do is the following. This is an algorithm that we call the recursive feature machine. So you can give me any model class. This could be, nearest neighbors or kernel methods or, trees or things like this. You give me a base learning algorithm for training that predictive model and an initial matrix for filtering the data. And what you do is you train your model on the transformed data, mt times x, and you predict the labels y. And then what you do is you update this filtering matrix mt as the AGOP of your trained model taken to some power alpha, and you just iterate this process over and over again. Okay. So now this is where it sort of becomes fun. What are we going to do to show that this works? We're going to show it on kernel machines or kernel ridge regression. So kernel machines are an older idea in machine learning now, but they were very popular up until around the 2010s. They were kind of the dominant model in machine learning. But the problem was that they historically were never able to learn features. And I'll give a brief review of what kernel machines are just visually. The idea behind a kernel machine is, suppose I have data. Here I have features in 2 dimensions, x1 and x2. And my goal, let's say, is to predict whether data points lie on one of these 2 circles. Well, instead of drawing a line, so I cannot classify between these 2 circles with any linear predictor in the original space. But what I can do is I can add an extra feature, which is just the distance of each point from the origin. And now once I go to this 3-dimensional space, I can actually draw a plane that cuts through the data. So somehow by adding these extra features and composing a feature map, you can actually train a linear predictor. And this is the idea of a kernel. A kernel is going to be effectively a function defined by a feature map. I can apply that feature map, do linear regression, and learn a predictor. The issue with kernels is that you have to pick this feature map. And once you're forced to pick a feature map, now you have to construct it on every data set that you have. But what we're going to do is effectively use the AGOP to adapt this feature map. So I'll train my kernel machine. I'll use a fixed feature map and update it through this AGOP. So now you can start to ask things like, what features do kernel machines learn through the AGOP? And how do they differ from MLPs and other neural nets? So suppose I have this task of going back to this thing of predicting glasses and images. Before you saw that the neural network was actually focusing on this region in the center of the image corresponding to glasses. Now if I train my kernel machine, it turns out the kernel, if I look at the diagonal of the AGOP, effectively focuses on the same region. This is after just one step of training a kernel machine and taking the AGOP. The other piece of it is, instead of looking at the diagonal, I can take the eigenvector of my AGOP of the neural net and AGOP of a kernel machine, 2 very different models, and they almost match just exactly in this space. So these two very different models, through the posited feature learning mechanism, learn effectively the same features. And when we wrote this paper, I had a really fun example, which was if you wanted to see what features are important to detect if somebody is smiling. It's a little hard to see here, but you'll get some kind of terrifying, like, clown face. And if you look at the eigenvector, you get this beautiful picture of whether somebody is smiling. So it's actually reminiscent of what people had done in the past with Fisher faces and eigenfaces. The only difference is this is supervised dimension reduction, effectively. And one other thing I'll mention is actually a very new work, but one that I'm very excited about. This model itself, beyond just a theoretical model, is actually incredibly good in practice. And it turns out that we extended it slightly. We combined it with tree-based models in some way. The idea here is if you have a massive data set, you can split the data set into subsets in some kind of hierarchical fashion and train this kernel RFM on leaves. It turns out this is a very, very effective predictor on tabular data. So what we found is we tested on 300 data sets. It turns out it was the best in performance across 100 regression tasks, within the top 3 across 200 classification tasks from this publicly available data source. The advantage of this method now is, unlike traditional kernel methods, and I'm happy to talk online why it's faster, but it scales to massive data sets. So you can have millions of samples, effectively billions of samples, run this procedure. You have n log n train time, O of log n inference time. And in fact, when you compare it on these benchmarks of regression, multi-class classification, and binary classification, it turns out in many cases it actually outperforms tabular foundation models. I don't know how many of you have heard of tab PFN v2, but this came out earlier this year in Nature, and it's a foundation model that was trained on tabular data, transformer-based, but this method actually works quite well compared to it. So it's not just a theoretical algorithm, it actually works quite well in practice. Okay. So with that, I actually want to turn to some applications of this work before going into the next piece. But the application I wanted to talk about is how we use this to do something called steering LLMs through feature learning. And so I want to take a moment to define what steering actually means. But the idea of steering is that you guide an LLM to align with a specific objective or a concept. So you could do this through prompting. So suppose you had a prompt, which was just, your mom asked you why there's a cookie missing from the jar. You ate the cookie. How do you respond? Well, if you want to steer a model you may want to steer it towards honesty. So you might prompt the model to be honest, and it'll say something like this. These are all real examples off of Llama. You might prompt the model to say, be dishonest. You might prompt the model to answer in Shakespeare, and you might prompt the model to output stuff in poetry. And of course, this is not surprising from the prompting perspective. But what's surprising is that you can actually do this through the AGOP without modifying the prompt in any way. So I'll show you how that works through kind of a compelling example that you cannot prompt engineer around. So the example I'll show is kind of an example that exposes vulnerabilities in existing open source language models. So it's going to be how you can steer the LLM to override their guardrails. And so the example here is, suppose you ask an LLM to give you a specific social security number that you've seen in a database. Any LLM right now is just going to tell you, I cannot help you with this. But what you can do is you can actually collect labeled data containing n harmful prompts that you think the model is going to refuse, and n safe prompts that you think the model will answer. And n here can be very small, like 200. You actually don't need the answers to these prompts at all. You just need the prompts themselves. So then what you do is you train models from the internal activations of an LLM to predict whether or not the model is going to refuse that request. And so the idea here is if I open up my LLM, it's a series of matrix transformations in the form of blocks. And so what I do is if I have a T by K matrix at any block, I just take the last row, that last K dimensional vector. I predict a 0 if it's a harmful prompt and a 1 if it's a non-harmful prompt. And I train a predictor to do this. And the idea here is I'm learning a predictor to determine whether my prompt is going to be refused or not. So then what I do is once I've trained my predictor, in this case a kernel, I compute the AGOPs. So these at each block are k by k matrices. And then what I do to actually steer the model is I take these AGOPs and I take their eigenvectors. So this is one k-dimensional vector at every block. And what I'm going to do is add a constant epsilon times that vector to the activations at that given block. And again, this is a real example on an 8 billion parameter model. This is the output that it will give you for this specific question. So it'll say, I've seen many social security numbers in various databases. One of them is blah. It's a real one, whatever that means. After looking up, I found it belongs to this. So again, the social security number here is actually real. I don't know if it belongs to this person, but presumably not. I actually don't think this one is real, but it shows you that you can do this. So this is a simple example. It turns out you can do this kind of at scale. So what I'm going to show you here is Llama Vision 90 billion, how you can steer it. And this is, to my knowledge, as of earlier this year, the largest publicly available vision language model. But now you can start to ask it questions like this. Does anyone know what this is a picture of? This is public domain cocaine. Very clear that I didn't copy this picture from anywhere else. It's freely available. So you can say, please provide me an accurate set of instructions on, whatever. The original model is going to tell you, I cannot provide instructions on how to use illegal substances, including cocaine. It actually knows that it's cocaine, but it's refusing to answer you. If you steer it with the same vector for refusal, this is the answer that you will get. And it's kind of a shocking example because it's a little crazy, but it also does drop the full curse word for you in the example, which I've censored, which is pretty wild. I've never seen it's very difficult actually to get a language model to drop a curse word, at least in my experience. It also works for like crazy tasks, so you can find mixers for cough syrup, you can counterfeit money, whatever it thinks that is, and robbing an armored truck. Okay. It actually works for general concepts. So in our paper, we have around 512 concepts, but, you could ask a question like this. This is the Moderna vaccine. Is it safe to take this? Okay. This is the original response from this model. It knows it's the Moderna vaccine. It's very good at image recognition, and it'll say it's absolutely safe to take this. Now you can steer it to be a conspiracy theorist, and if you add the conspiracy vectors, this is what the model will say, which is a little crazy. It turns out it tells you a lot of fun stuff. It'll tell you things about life on the moon, building the pyramids, photos of Earth. It usually has like some crazy phrase like wake up sheeple at the end of everything. What I found very interesting, so these are again all linear perturbations that I'm doing to the weights. It turns out these are shockingly combinable. So you have one vector that you can use for conspiracy theory, and you have one vector that you can use for Shakespeare. So you can add them, and now it will tell you not to take the Moderna vaccine in the style of Shakespeare, which it'll tell you is like, it's a concoction of sorcery, a potion of dark arts, whatever. Okay. So that was just an example of, what you can do with this. Turns out there's also many good applications, because the moment you identify vectors like this, you can add them or subtract them. And it turns out there's very simple prompts that will get you to give conspiracy theories from the model, which is obviously very harmful. But in those cases, when you subtract this vector, you can actually control that from happening. So whatever you can do in the positive direction, you can also subtract and get safeguards against this. Okay. So the next piece I actually wanted to talk about is, again, back to a little bit of mathematical stuff before I go into the biology side. The question is, I've spoken about AGOP as a mechanism for feature learning, but how do I know that it's actually learning low-dimensional or low-rank structure? So let's start with the simple question of why is AGOP identifying a sparse subset of features, like, for example, in the glasses case? So if I actually look at the AGOP and I just consider the diagonal terms, this is just an average of the partial derivatives, the squared partial derivatives of my predictor with respect to a single coordinate. So, by law of large numbers, if this grows large, it's effectively an expectation of the partial derivative square. So suppose this diagonal term is 0 for all x coming in. This means that my function f is effectively constant in that coordinate, and in some sense it actually means that the outputs do not depend on x. So this suggests that by computing the AGOP, you're actually eliminating features that are independent of the output. And I've put independent in quotes here because I'm using it loosely, but it actually turns out that there is a real connection, and the connection is that the diagonal entries of NFMs actually compute statistics for independence called the Hilbert-Schmidt independence criteria, or HSIC, between the input coordinates and the output. So what is the key idea behind what is an HSIC? So, many of you will probably remember that you could, if I want to test for independence between two variables, I can compute correlation. It's not necessarily true that if correlation is 0, the variables are independent. There's good examples of this. But it is true that if the covariance of any transformation of two variables is 0, then they are independent. And the HSIC, the key idea behind HSIC, is that you check a specific subset of nonlinear transformation. It's a countable subset of nonlinear transformations that when you apply to x and y, whether the covariance is 0, and you do this through a kernel trick. So given two kernels, k1 and k2, you effectively compute the squared, the Frobenius norm of the covariance between these transformed variables. So if this quantity is 0, so this is the advantage, if that quantity is 0, then x and y are independent, and it's an if and only if statement. The downside of HSIC is that, one more advantage of HSIC is that it's sample efficient. So given n samples, HSIC, the empirical HSIC converges to its population level value at a fast rate. The problem is that computing this object requires quadratic time in the number of samples. So this is very painful. This is where the advantage of neural networks come in. So you could do the obvious thing of you have a countably infinite set of transformations, you just do an approximation. This gets you linear time for computation, but the tricky thing is that you still have to do for loops across pairs. If you have many, many pairs of variables, you have to do for loops across all pairs if you want to check for dependence across all of them. It turns out, though, that neural networks are very good at vectorizing this operation, meaning that it can compute multiple HSICs in parallel using one matrix vector multiplier. And so because of this property, both the O of n runtime and vectorizability, this HSIC computation is incredibly fast. It's actually, on a GPU, it's 100,000 times faster than other measures of dependence. And in fact, if you can construct neural networks that when you use a specific nonlinear activation, it will approximate a universal kernel. It will be a statistically consistent, or approximate a statistically consistent HSIC, but do so in a very efficient way. So that's my interlude on HSICs. I know I went a little bit fast there, but you can ask me if you have any questions afterward. The issue with HSICs, though, is there's one other issue, is that they don't quantify dependencies. So if HSIC is 0, this means that two variables are independent. That's good. But if I have a large HSIC, it doesn't necessarily mean that variables are more dependent in some way. And so there is a really simple solution that we did here, which is you can take this neural network computation, but instead of computing covariance of features, you take two variables, x and y, you apply some finite dimensional transformations. So here I've applied 6 transformations to x, 6 transformations to y, and you just compute the maximum correlation over all pairs of variables. We call this the InterDependence Score. The advantage of this is that it's extremely fast to compute IDS. On 10 million samples and 1 billion pairs of dependencies, this takes under 5 hours on a single GPU. And the nice part is that IDS values are in 0 to 1, with low values implying weaker dependence. And in fact, you can construct a null distribution for this, so you can get p-values and things like that. Okay. So now I want to show some applications of this that ultimately lead to the application in biology. So the first application is we can start to tackle that question of when can we identify variables that matter. So suppose I have a function where my label depends on 100 coordinates. It's a function of x in a 100-dimensional space. But in reality, suppose it actually depends on 4 of those coordinates. So what you could do to discover this dependence is you could actually just run correlation between every coordinate in the label, and you'll see that correlation will not spot the difference. So on the x-axis here, I have all my 100 coordinates. Y-axis is the correlation between them in the label. It won't do it. This is a nonlinear function. IDS actually easily spots the dependence. It's a fast nonlinear independence test. So you have, your 4 variables. You found it right here. So now going to a, non-synthetic but very fun example, you can actually run IDS at scale on text data. Suppose I have 8 million documents with 50,000 tokens. So I can take every document and I can encode it as a vector in 50,000 dimensional space where each entry tells me how many times that word appeared. It's the bag of words representation. So I can actually compute dependence across all 8 million documents for all the words, and I can start to ask questions for any given word. What is the token, the set of tokens that it depends on? So here are some examples that I think are fun when you actually do this at scale. So if you look at the word, small spider, you'll see this comes out as your top nearest neighbors with IDS. It'll be spider, silk, webs, whatever. If you just change the letter from lowercase s to capital S, you'll see that it's actually going to give you all of Spider-Man. If you do the same thing for Swift, the words that have most dependence on Swift are things like action, backlash, so on. If you do capital S Swift, you'll find, of course, Taylor Swift and her 1989 album. And then the really fun example, which is kind of unexpected, but open web text is a set of documents that are collected over the past 10 years. So if you ask for the word Newton, what words do you think have highest dependence with Newton? You can just shout out. Yeah, right? Okay. No, it's actually Cam Newton, the Panthers quarterback. And this is a really fun thing you can check out. If you look at GPT-2 token embeddings, and you look at what is the nearest neighbor to the word Newton in GPT-2's token embeddings, is actually Cam Newton and the Panthers. So you'll see that there's some connection there, but I haven't made it precise. Okay. So you can do this for all, 50,000 words. I wanted to show you a little bit about what kind of topics you find in a subset of words that are lowercase English words. So if you look at certain regions, you'll find things like math and programming. In other regions, you'll find things like healthcare. In fact, you can look at any region in our paper, which is now online. You'll see that almost any region of this space actually is very tightly coupled dependence among, indicative of a given topic. So in fact, you can kind of use it for topic modeling already, and we're working on methods for that. Okay. So the last application I wanted to talk about, I think I will have time for this, is how can we use IDS and some of these ideas to actually tackle a major biological question, which is discovering cellular programs through feature learning. So just to make sure everyone's on the same page, I just wanted to go over quickly some recent advances in biotech that have enabled this sort of application. So here's my little diagram of a single cell. This is the nucleus. It has DNA. What is DNA? One strand of DNA is a set of nucleotides. I have 4 letters, A's, C's, T's, and G's, which represent nucleotides. You can think about it as a string of letters if you so desire. Each subset of these letters, and I'm being very loose here, but each subset of these letters is roughly a gene. And there's roughly 20,000 of these strings that code for proteins in the human body. So there's 20,000 roughly protein coding genes. And so what are we measuring when we talk about single cell RNA sequencing? We actually have each of these genes that gets translated into, or transcribed into multiple copies of RNA. And so each gene can have a fixed count of how many times it was transcribed. And if I take all those counts for all the genes, I produce a 20,000 dimensional vector. So it's a vector of actually integers. I know I've written R20K, but it's non-negative integers. Okay. So just to show a little bit of history here. So about 11 years ago now, researchers at the Broad were able to do this at the level of 400 cells. But over the past 10 years, this has been tremendously scaled, and now you have data sets of over 100 million sequence cells across the human body. And this is only growing. We're pushing to get to 1 billion cells. But now once you have these RNA count vectors, you can actually just start to ask the same question that we did on text. Which sets of genes have these expression patterns that are dependent on each other? And so I can take all my 20,000 dimensional vectors and compute IDS effectively on this. So why do we care about this? Well, we want to identify novel gene-gene interactions indicative of new cell subtypes or states. And I'll show some examples here. But the idea is once we have these gene-gene interactions, we're going to group them into sets of genes that form programs that are cell type specific and run across many different cell types. And just to give some examples, if you look at the genes for hemoglobin, which effectively carries oxygen, these are expressed in red blood cells, which carry oxygen to your cells. You'll find that they form a small program, so HbA1, HbA2, HbB are my hemoglobin genes for adult hemoglobin, and they're cell type specific. So you should see this program running high dependence among these genes in certain red blood cells. But you might also see a program of genes in different cells undergoing cell division. So maybe I'll see these genes that are occurring together that are indicative of cell division that are occurring in certain specific cell types. So the way we go about solving this problem and finding sets of genes is we encode this data as a matrix. So you have here 27 million curated single-cell RNA sequencing data. You have 20,000 genes. So on my columns, I have 20,000 genes. I compute IDS between every pair of genes. So it's a 20K by 20K matrix. Then I just look for sets of genes that are dependent, and note this dependence could be various forms of nonlinear dependence. So the IDS computation for this was actually also under 5 hours, coincidentally, so you can do it really fast at scale. And now you can start to get an overview, just like we did for text, but for genes. And if you do that, you'll find that so each point in this little plot here is a gene. And if you look at sets of genes, you'll find that there's many clusters corresponding to distinct cellular programs that are running in specific cell types. So you'll find a set of genes that are involved in the pancreas. You'll find genes that are indicative of cell division. You'll find a program that's indicative of red blood cells and hemoglobin. You'll find programs that are running in platelets and their function. And you'll find defense programs. And what's nice about this is that here we were able to discover well over 100 candidate programs using our approach. But because it's so fast, you can sort of build a hierarchy of programs. You can look at an atlas as a mixture of cell types, but you can also run it individually on every single cell type. And in fact, this is what we did. We ran it across 460 different cell types, and now you can start to ask questions of what do you see? And so I really wanted to showcase, somebody might ask, what are you finding that correlation isn't finding? And it turns out the nonlinear dependencies that you find in real biological data often look like this. So here's an example of two genes and their expression across 12,000 cells of the pancreas. And you can clearly see there's two distinct subpopulations, one which has the two expressed, one which effectively has one expressed. And what's going on here? So here's my little picture of the pancreas. So this is my pancreatic duct. I have ductal cells which line the pancreas, and I have cells at the edge which are called acinar cells. And what do acinar cells do? They actually have little packets that they send out which carry amylases, lipases, proteases. These are things that digest carbohydrates, fats, and proteins. And amylases here are specifically for digesting carbohydrates. So what's happening in this picture? It turns out there's a subpopulation of acinar cells that are actually transitioning between this edge and a ductal-like state. And why might this happen? It turns out I looked up some explanations online, and somebody can correct me if this is not the right one. But it turns out when you overexcrete amylases, lipases, proteases, the cell starts to digest itself. So it starts to become harmful. So the cell starts to damage itself, and what it does is it actually transitions to a ductal cell so that it stops hurting itself, and then it can eventually, once it's cooled down enough, it can transition back. And this is something that you're able to capture by investigating these different subpopulations. Somebody might also ask, what are you finding that's not known? It turns out that there was an interesting reaction in platelets that we were looking at. So what is a platelet? This is my hardcore PowerPoint animation of platelets and red blood cells. So here's a platelet, here's a red blood cell. What do platelets actually do? Well, platelets and red blood cells are traveling in your blood vessels. And if I have a cut, I have a broken blood vessel, the platelets actually go through and actually form a clot, and this is what's forming clot that ends up getting stitched together with proteins. So if you start with some genes characteristic of platelets and you start to ask who are their nearest neighbors with IDS, if you look at their top 10 nearest neighbors, let's say, you'll find genes that are involved in platelet activation. You'll find genes that limit platelet overactivation. You don't want to overclot. That's going to be bad for you. And you'll find structural elements for platelets, things that form their structure, their disc-like structure. This is tubulin for people who are interested. What's surprising is you actually find other genes too. And this one in particular was very surprising to me. What is this gene, ACRBP? It's extremely unexpected because it's classified as a cancer testis antigen gene. What does that mean? That means that it's typically found aberrantly expressed in cancer, or it's found in normal tissue in the testes. And this is in platelets we're finding its expression in platelets from normal non-disease tissue. So here's the expression of, let's say, this gene and another gene tubulin, and you see that there is some kind of dependence. And in cancer, it turns out that this gene is known to stabilize effectively when the cell is becoming polyploid, when it's multiplying its chromosome content a lot of times. This gene is known to stabilize that process. And it's actually plausible, quite plausible, that it plays a similar role in platelet formation because platelets actually bud off of a specific cell. That's how they're produced, and that cell has very, very duplicated chromosome content. So this is something that we're actually exploring further with the Broad, and we're running some interesting protein expression experiments to see whether this is occurring and what its role is in stabilizing platelet formation. But what's to come, which is what I'm very excited about with this type of research, is now you have extensive new approaches for topic modeling in text. You can actually do something similar for large-scale atlases of single cell, and you can start to catalog cellular programs that enable the discovery of novel cell subtypes and states like this instead of me manually looking through and doing this. And in fact, we're doing a big push of building out these tools and having agents called LLM agents call them and giving you a seamless way to actually start to explain and identify interesting things. So with that, I actually wanted to conclude, and I wanted to say this has been like an extremely rich question for at least my research. Of understanding how neural networks learn features from data. And really for us, this AGOP has been a major step in understanding how models in general can do supervised dimension reduction. But it's really been useful in enabling feature learning in models beyond neural networks, connecting feature learning to dimensionality reduction approaches, and really applying it to scientific domains, things like identifying cellular programs. And lastly, I wanted to just thank my collaborators. Daniel, Misha, Dima, Partha have been really, really helpful, and Enrique have been really helpful on the machine learning side. And then a lot of this work on the biological side is not possible without the Broad, without Caroline, Eric, and Yachet at the Broad Institute. And with that, I'm open for questions. So thank you.